The overall goal of the following experiment is to utilize the density of mapped read positions from chromatin immunoprecipitation sequencing data to estimate the posterior mean read density across the genome. This is achieved by pre-processing the mapped ChIPseq reads into blocked density profiles with the same number of reads falling within 200 base pair non-overlapping bins. Any adjacent bins with the same density are merged into a larger block. As a second step, posterior mean densities of each block are calculated recursively within the context of all surrounding blocks using a Bayesian model with forward and backward filters, where the read count for a block is modeled with a Poisson distribution with a theta parameter that takes on a gamma prior distribution with alpha and beta parameters. Next, posterior mean density estimates of each block are evaluated for significance based on whether or not it exceeds the 90th quantile with respect to the input control background density in order to generate the final enriched genome segments. Results are obtained that illustrate the progression from raw sequenced reads to posterior mean read density estimates, and finally enriched islands on ChIPseq data during BCP analysis. Furthermore, the results show that BCP outperforms a competitor tool, ChIPseq.
The main advantage of this technique over existing methods, like ChIPseq, is that BCP used the most recent advances in hidden Markov models, so it better characterizes the nuance of ChIPseq data analysis than previous heuristic methods. This method can